So thanks very much for having me. I'm excited to be here. Today I'm talking about graphical models and a project inspired by a line of work and collaborations on functional neural connectivity. So to get started, over the past decade, there's absolutely been an explosion in the type of data we're getting from neuroscience technologies that can measure nearly every aspect of the brain. And this is one of the great mysteries, right? It's no longer our genotype, our genome. It's really like what's going on in the brain and demystifying this. And so these technologies can really help us understand things, but there's many open questions. And so what I'm going to talk about today is motivated by a really big, huge collaborative project. This has been a 10-year project that they've been working on called the Microns Project. And the goal here was to understand a millimeter cubed portion of the mouse brain that fully contained V1 in the visual cortex of the mouse brain, and to get structural and functional information on all. It was about 200,000 total cells and about 75,000 total neurons. And the data that we're interested in are two modalities of the data that was collected from this project. And I should mention this project is running at about $150 million funding effort to collect this data. So this was a massive undertaking, and it's still ongoing. And the two modalities that we're interested in are the structural data, so right here, and this is collected from EM microscopy data, which is really taking high resolution images of the structure. And what you're seeing is individual neurons. This is the soma, or the cell body of the neuron, and we have some dendrites and axons extending out that, of course, form the synapses. And at the same time, we have all this structural data from EM microscopy. There are functional recordings of the neurons while the mouse is alive, running on a little treadmill, watching movies. And so we get all these functional recordings, and this is kind of a visualization of the type of data you would get, but you basically get, for each neuron, we get activity traces here of all of these all of this data. So this is a massive amount of data. It's for 75,000 neurons in the mouse visual cortex. The calcium imaging data is how we get these, the functional data, the neuronal traces and the activity traces from 11 different sessions, and electron microscopy for the structural data. And this is the largest multimodal connectomics data set of its type in the mammalian brain. So this is a really exciting data set, and really opens the door for answering some really big unknown questions in neuroscience. And so what motivates my work today that I'm going to talk about is asking, how is the brain functionally organized or connected? And how does the brain's function and functional connectivity relate to the brain's structure? And so this has been addressed on very small scale studies in small animals, typically not mammals before. This has never been done on a large scale in the visual cortex of the mammalian brain. And data like this multimodal data, is the first of its kind that can allow us to do this, okay? So just a little bit about, I'm not going to go into details about calcium imaging data, but calcium imaging data is a video recording of the neurons as it's firing in the active brain. And each session is actually an hour-and-a-half session where there's all these different experimental protocols that the mouse is watching. And actually, let's just attempt to, here we go. Wait, I don't want to do that. There we go. Let's just see a quick video, actually, of what is going on. And so this is an actual video of what you get. This is called a mesoscope from 2-photon calcium imaging. You see the cortical structure in there, and you see the neurons fluorescing. The lights are a little weird here, so you can't really see that well. But you see them. This is like a slow motion version. You see them lighting up. And now this is their neural tuning, basically like what type of visual cues they respond to. And so they're color coded according to that. And you can kind of see them fluorescing and firing. So what happens here in this video, and you can see that the study was trying to capture all of V1 in a millimeter cube of the mouse brain. So you can kind of get a sense for what that data is like. And let's go back over here and see if I can get this set up again. Okay, great. Okay, so so we get this calcium imaging data, and there's a ton of pre-processing that goes into it to go from this video recording of each neuron firing down to what we call neuron activity traces. And these are for each neuron right here is a particular row of this. This is basically when this neuron is firing. And what happens in this technology is every time a neuron fires, it intakes calcium. And so you're seeing the calcium delayed response of every time a neuron's firing. So we can't see individual spikes from neurons, but we can see largely the activity. And so we see this burstiness in the neuron activity traces. And so there's a lot of pre-processing that goes into the data to get it in this form. And including, we do some whitening also, so it should be hopefully mitigates the temporal dependencies that we have here to get data that are neuron activity traces. And this is the type of data we're going to work with when we talk about functional connectivity. Okay. So functional connectivity, what is this? This is just the statistical dependence amongst the neurons based upon their in vivo or live brain activity that's going on. And there's several types of connectivity. There's two main ones, dynamic and intrinsic. Dynamic is just like the word says, it changes over time. And so this is the functional connectivity that might change for different stimuli that the mouse is doing. What we're interested in, our goal is to try to find how the functional connectivity relates to structure. So we're going to be interested in the intrinsic connectivity, and this is the connectivity that does not change over time and that is static regardless of what the mouse is doing and what the stimulus is. And that's what we're going to be interested in. So specifically, each neuron here, each soma is a cell body, we're going to represent as a node in a network. And so each neuron's the number of nodes or the number of neurons that we have in the study. And our goal is going to be, unlike synaptic connectivity, which is actually the structural connectivity that's observed from the EM microscopy data, our goal is going to be to infer an edge between these neurons if these two neurons are communicating with each other in the active mouse brain. Okay, so that's our inference task here. And there's a lot of existing approaches, including from the stat community. In neuroscience, actually, what most people do is typically, I would say 90+% of papers are really simple metrics of like noise correlation is most common. They basically adjust for the stimuli, and then they just look at the correlation between two neurons and say they're connected, they're functionally connected if the correlation is high. So the stuff in the neuroscience community is really simple. We've tried this. There's basically no relationship, and previous literature has not established any relationship between noise correlations and synaptic connectivity before. Go figure. It's just correlation. Kind of, if you look at these brains and these neurons, everything is correlated, right? And so we need to go a step beyond that with a much stronger form of dependence with graphical models, and that's going to be the approach that we're going to explore today. Okay, yeah, question.
I was wondering, I think the question is intrinsic connectivity, but regardless of what the mouse is doing, why use a different kind of stimulus on the mouse instead of getting the data at rest?
Yeah, that's a great question. So these recordings that were done on this mouse are an hour and a half, and they have some of everything. They do have resting state is what you call an fMRI. In this type of data, it's called spontaneous activity. And so we do have about like 10-minute increments of spontaneous activity, but that's simply often not enough data. And we want, actually, during spontaneous activity, the visual cortex does not fire that much. And so it's really important to have visual stimuli that stimulates the visual cortex. Otherwise, you simply don't see firing of neurons in the visual cortex. So the experiment pattern, and I actually worked with them to help design this experiment, which is kind of fun. They actually do they had increments of like Hollywood movie clips, and then they had these like natural images that were shown, and then they had great visual gratings, which are very typical in mice, but they also had this like three-dimensional, and they developed this like mouse virtual reality. If this mouse was like running through a maze and like this hawk comes down and swoops down and it was actually kind of fun. It's like a mouse like video game. And that was what was done in the experiment. And it was all to just elicit as much activity from the visual cortex as possible, was the goal, right? Okay, cool. Okay, so we're going to model this with graphical models. And these are just multivariate probability distributions where the dependencies can be denoted as a network. And specifically, we're going to work with undirected graphical models or Markov networks where each edge is a conditional dependence relationship. And so we might look at this little triad here and say, we would read this as A is conditionally independent of B given C. And if C is really kind of driving the activity we see in A and B, it's very likely that A and B are also really highly correlated. And this is what we see, of course, in neuroscience data. Like the neurons are very, very highly correlated, even adjusting for stimuli. But what we want to ferret out is kind of, you know, which neurons are really potentially driving that activity or communication amongst neurons. And so graphical models are a really powerful tool that allows us to do this. Okay, so our inference task is from data that we've observed on P different neurons. We want to learn the edge structure between each of the neurons, and so learn the graphical model. This is often called structural learning in graphical models. And just as an example, this is super widely studied. Gaussian graphical models, we assume that the data is multivariate Gaussian. And of course, the precision matrix or inverse covariance matrix lets us read off the conditional dependent and independence relationships. In particular, if xi and xj are conditionally independent, then theta ij is equal to 0. And so this is a fantastic model because it takes our inference task of edges to simply getting the 0 pattern in theta. And there's tons of fantastic, great models to do this. Many in this room, of course, have probably worked on some of these. There's probably like 10,000 papers on the graphical lasso. So everybody is probably pretty familiar with this. Or has at least heard about this. And this is just sparse maximum likelihood estimation. The first two terms are just fitting the multivariate log likelihood. And then there's a sparsity penalty on the off-diagonal elements, typically of theta. And there's many other ways to estimate this for Gaussian graphical models. Again, this stuff is like super well studied. So lots of examples here. But our goal is to learn the intrinsic functional connectivity amongst 75,000 neurons. And this is going to present a lot of challenges here, in particular because of how the data is collected, the properties of this data, and also the sheer size of this data. So a little bit of a story. When I first got involved in this collaboration, this is probably now like 7 years ago. I don't know exactly when it was, but several years ago, my neuroscience collaborator, Andreas Tolias, was the one that started talking to me about this. And he was like, Genevera, we're going to be collecting all of this data and you do graphs, right? And he was just like, can you do some graph stuff on my data and get me a functional connectivity graph? And he was like, can you get it to me in a couple months? And at first I was like, okay, well, maybe, like, tell me about your data. And he starts telling me about it. And I'm like, oh my God, that's like a multi-year project. And we need to write like 3 grants to do this. This is really hard. And he was just like, no, it's not gonna be that hard. And I started explaining it and he was like, okay, it's that hard. And he was like, well, you need to hurry up because we're gonna have the structural reconstruction of the graphs before the functional ones. And ironically, now they're still working. We've got estimates of the functional graphs. They're still working on the structural ones. They think the structural graph is about like 3 to 5 years out because it's a big computer vision problem. So anyway, kind of a fun story. So we've been doing a lot of work related to this. So my approach to this problem was basically like, wow, this data is a lot more complicated than typical scenarios where we apply Gaussian graphical models. So what I've decided to do is like take each like major challenge and try to like think about it very thoughtfully and write a paper addressing and develop a method for that particular challenge and then eventually put everything together. So that's really been our strategy. Today in this talk, I'm just going to talk about 3 of these areas because they're kind of 3 interconnected projects related to non-simultaneous recordings, latent effects, and also dealing with the sheer size of the data. Okay, so let's get started with graph quilting. So again, the microns data has calcium imaging data, 75,000 neurons, millimeter portion cubed of the brain. And here's our little like brain cube that we have. But there's a problem. There's absolutely no way from the physics of the technology of 2-photon calcium imaging to image 75,000 neurons at once. So there's two issues. The first issue is that the temporal resolution, the number of neurons you record, is such that if you record tons of neurons, tens of thousands of neurons, you don't get sufficient temporal resolution to see spiking activity behavior. Well, that's a problem because our whole point was to see like when neurons fire. The second issue is a depth issue. This technology can only do like image thin slices in the brain and can't do a 3D portion of the brain at all. So we can get no depth whatsoever in the brain from a single recording session. So because of this, the experimental design and the data was collected like this. We've got a brain cube, and here I'm showing the samples. Just think of like sessions that we have by neurons. And the brain was chunked up into slices, these vertical slices of the brain. And there was one recording session, and then we go down and down and down until it looks like this. So we kind of go down in 11 different recording sessions in these slices in the brain going down. But of course, this creates a really, really big statistical challenge. Look at this. This is our data. This is our observed data like this, and this is our unobserved data here. And of course, we're interested in connectivity, which means we need pairs of neurons that were observed together, or pairs of variables that are observed together. And of course, for the pairs that were observed together, they have this quilting pattern, and hence we call this problem a quilting problem. Someone told me that this was a misuse of quilting, and it's really like stitching or patching or whatever. I don't, okay, whatever. Quilting sounds better. So we call it quilting. But the issue is like there's all these unobserved pairs. We call this O complement. And in O complement, there is no joint data whatsoever on these pairs of neurons, right? These are variables that were never observed together. And so the question is, can we construct a graph not just for the data that we've observed, but for all the neurons in the entire experiment, even if there were no joint observations? And in other words, can we learn a graphical model from just partially observed interactions or partially observed covariances? And another way of phrasing this is it possible to infer conditional independence when you can't even estimate marginal dependence? Think about it. We can't get a correlation. So how are we going to get conditional dependence, which is like a higher order up? So this seems like a completely impossible problem. My neuroscience collaborator was adamant that we solve this because he, oh yeah, question. Yeah. Yeah.
Would you be able to infer marginal dependence using the fact that it's correlation
oh, and just do a positive definite completion? You could. We explored that right off the bat. That was the first thing we looked at. The issue is most of those positive definite completions assume low rankness, and most graph structure is not consistent with the low rank assumptions. And so what you end up getting is you can infer the graph well here, but you actually cannot. So far, we're still working on it. We don't think that there's easy provable guarantees in O complement at all using that approach. Yeah. Yeah, I mean, you can try covariance completion or correlation completion approaches, but the guarantees that are out there for these approaches assume essentially missing at random, and this block missingness violates all the assumptions. So then that's why it doesn't work, right? And you can also show that most graph types, unless they are exactly like have separated connected components, they're not low rank. And so you basically can infer only connected components in a graph. You can't infer the whole graph. Right. So yeah, the great question. Yeah. So when we started this problem several years ago, I was working with a postdoc at the time, Giuseppe Vinci, and a collaborator, Gautam Dasarathi, and we both were thinking about this, and we spent like, we thought about this for a while, and we were trying to tell our neuroscience collaborator that it was impossible to do. And we started on many lines of proofs about an impossibility result. But it turned out, of course, we eventually found a mechanism. And one of the things we did try was this kind of correlation completion approach, but it wasn't quite, didn't quite work out. But we did find another approach that worked. So let me kind of outline how we're going to go through the results. And what I'm going to do is I'm going to show you the intuition behind our setup and how we got these results. I'm not going to go into details about them. I can refer you to the paper. So what we're showing here is this is our true edge set E. So this is the true adjacency matrix of our graph. So this is neurons by neurons here. And the black dots clearly represent edges between those pairs of neurons. And this will be our observed variable pairs. So this is our observation pattern. And in gray is O complement pairs that we have not observed together. So our main results, and this is a population result first, we extend this to finite samples later. Our main result says it is possible to identify the graph perfectly with high probability in the observed portion. And in the unobserved portion, the graph is not identifiable. The best you can do is find a superset of the edges, and we can identify the minimal superset that covers all possible edges consistent with the observation pattern, and that this is the best you can do. And this is kind of our main result. And I'm going to walk through the intuition for how we get here. So we start off with the MAD-GQ estimator, and this is just, we call it this from the max determinant covariance completion of Dempster. So yeah, we are using some type of covariance completion here, Daniela, to answer your question, but this is a very specific one because this assumes in O complement that everything is zero, erroneously. We know there are edges in O complement, but we're just going to assume that there's no edges for now and just do maximum likelihood for the partial observed portion of the data. Okay, so we're going to erroneously assume this. We're going to call this estimate theta tilde. Again, I'm showing all the main results in the population case, but of course, these are extendable to finite samples. So the first result is if you apply the MAD-GQ estimator, something really cool happens. You can show that there are no false negatives in O at all. So the observed portion only admits false positives. You're not ever going to miss an edge. Okay, so this is a really big deal. We're not going to miss edges in O, which is important. So after staring at a lot of these graphs a lot produced by the MAD-GQ estimator, I noticed, wait a second, all of these false positives that are shown here in green, they're really kind of small in magnitude. And it turns out under certain assumptions, you can show that there exists a hard threshold operator that if the false positives under these assumptions are bounded away from the minimum edge strength in the graph, you can simply lob them off with a hard thresholding tau and get perfect graph recovery in the observed part in EO of the graph, okay? So suddenly we've got perfect graph recovery here by simply hard thresholding a very simple partial likelihood estimator. And then in O complement, we have lots of false negatives, of course, that we need to deal with, okay? So the intuition here is, so O complement is a lot more difficult, right, to solve. And the issue here is that you can show that O complement is not identifiable ever, meaning we cannot recover the edges there. Yeah, Armin.
Why is it that, you know, where did the power come from and what the made to have small?
Yeah, great question. So the type of assumptions we need are that the effects that we have, kind of the maximum effects, this is actually a very similar assumption, which is where I'm going to the latent variable stuff that we were talking about earlier. Yeah, and it's basically disentangling the effects from the latent variables. Essentially, if there's an edge, if like right here, there's edges, let's go back one. If there's edges right here in a node that were unobserved, it's going to produce false positives here that are typically small in magnitude if there are other pathways to get to that edge from this edge. Like, so it has to do with it. It's kind of a graph connectivity result. And you can map them through the Schur complement to get them. But I can answer in more, we can talk later about some more of the technical stuff. But yeah, in general, this mostly seems to hold for like, the graphs need to be sparse and they work really well for more like small world graphs where there's lots of paths between nodes and sparse graphs, but dense graphs are totally screwed. Like this result will not work. Okay, so the issue in O complement is it's totally not identifiable from the data that we've observed. And so the best we can hope for is a minimal superset that contains all possible edges in O complement. And so what we do is we have this like clever use of Schur complements for the different patches of the data that are observed that gets us this minimal superset. And this minimal superset means there will absolutely be false positives in O complement, but we're not going to miss anything. There's not going to be false negatives. And by minimal superset, we mean this superset is as small as possible. So let me go through the intuition for how we construct this superset here to really understand this in like a simple scenario. So this is an example of a kind of tiny but small world connected graph, okay? And this is the estimate we get from the MAD GQ, where of course we've got false positives in O and false negatives in O complement. And I literally stared at so many of these. I had Giuseppe just like print out MAD GQ estimates for like oodles of graphs. I kept staring at them and I was like, wait a second, these false positives are not random at all. And actually, this was the proof technique we ended up using. We showed something really cool. A false positive can occur at this point in the graph if and only if there is a missing edge somewhere in O complement connected to this node. We don't know where, but we know it's there. And a false positive can occur here if and only if there's a missing edge on this node neighborhood somewhere. We don't know where. Again, same thing here, if and only if, and if and only if. And suddenly we're left with a Cartesian product of 4 possible edges in O complement. We actually can tell you that there's really only 2 edges, or 2 edges is the minimal that covers it, but we don't know which of the 2 it could be. It could be these 2 or these 2, right? We can flop them. And so these are then our false positives that we get in O complement, and that's how we construct the minimal superset. So again, just to walk through this is our MAD GQ estimate right here, which doesn't look very good, but then we can apply hard thresholding to O and this superset method to O complement to get this minimal superset. Yes, Holly.
There's an assumption there that basically the effects don't cancel each other.
Yes.
There's also a faithfulness.
It's not quite a faithfulness assumption, but it's pretty darn close. And all of these results are almost surely or with high probability because there could absolutely be a boundary effect where the effects exactly cancel each other out and you get a 0, but that's like a probability 0 event. So it is possible to occur. The assumptions that are needed to get this superset, they're like these path assumptions, right? If there is not an alternative path between the nodes, we can't detect it at all. So if there's 2 nodes and there's a missing edge that's connecting them in O complement, and there's no other path between those 2 nodes except through O complement, we cannot identify that edge in O complement. So that's a very simple way of saying it, but it has to do with these paths that go through O complement connecting them. And that's why we're fairly confident that we can recover graphs really well that are sparse, but are still like, there's lots of paths through them. Like an Erdős-Renyi graph is actually kind of hard sometimes, a sparse Erdős-Renyi graph, because there's not enough paths between the nodes. But a small world or a scale free, there's lots of potential paths and it's much easier. Does that sort of make sense? Yeah. Okay. And also, even though we call this graph quilting, you can totally do this in, I don't know what this is, like plaid models or something like this, like generic observation patterns. And so this could be potentially useful for many other types of data as well. Okay, so just to summarize, we show the necessary and sufficient conditions under which we can recover the graph in O, and we can cover the minimal superset in O complement. And we also develop an algorithm that does this in finite samples and recovers with high probability and finite sample estimators. So the sample complexity is not great. It's the minimum sample size for any of the patches there, but we do think that that can be improved in future work. Okay, so just showing you kind of a picture here. So this is I mean, we want to validate these results, but we don't know the true connectivity graph for neuron data. So this is actually kind of a real data inspired simulation by these are real neurons. We estimated a graph from this, and then we regenerate a Gaussian graphical model under these different observational schemes with 20% missing and 40% missing edges. Okay. And here what we're doing is showing the area under the curve of exact graph recovery versus the sample size n. And the solid line is what you would get with the graphical lasso. So the solid line is assuming all the data is observed, and the dashed line is 20% missing and then 40% missing. And you can see like, we do take a hit with more missing, as you would totally expect, but it's like fairly close and we're doing fairly well compared to observing all the data. And so the experiment was done in these kind of horizontal slices, but we're showing you vertical slices here, but then oriented horizontally. So it's a little confusing. This is the graphical lasso. What you recover, you definitely see that kind of small world structure. This is with 20% missing and 40% missing. And you do see there's false positives and false negatives, but overall the graph structure is still preserved. You still see some of the major structure in the graph there. Cool. Questions? Yes, please.
Yeah, like you're doing like renewing edges for it. Can you talk about how this is not an area streaming graph?
Yeah, okay. So this is so our results are totally about support recovery of the graph. This is really important. We cannot recover the edge weights at all, and we cannot recover theta. So we can only recover support recovery. We don't think it's possible using this model. We know it's not possible to recover theta at all. I think the only hope of recovering theta is by extending the existing, like Daniela was saying, covariance or correlation completion methods so that they work in this block missing setting, and developing some type of consistency there. I think that's the only hope. Yeah. Yes. Oh, you're totally right. Quilted part, we can actually get theta. It's the O complement. We have no hope of getting theta. Yeah. That's right. Yeah. We can't disentangle. Yeah. Yeah.
So just like as a statistician working with the scientists,
yeah.
Right? So the kind of interesting thing about this type of result, so you sort of like a point estimate for the support. But you have very different like confidence in the sense about one set of the object that you've estimated, and a different type of confidence about a different set. Like how do you communicate that, and like how do the scientists wrap their head around that?
That's a really great question. I was going to go there later. I'm not going to jump in my slides now, but I'll come back to it if I have time. But yeah, so a direction we're going now and pursuing with my fabulous postdoc, Lily Zheng, who's on the job market, is actually doing uncertainty quantification for different parts of the graph based on different sample sizes and what's observed. So yeah, that's, I think, a super, super important part of the equation and something we're working on. Turns out to be really hard in this setting, but is critically important. Yeah. Cool. Okay. So let's jump ahead to latent variables. Okay. So here's the deal. We've got this brain cube. It's got like 75,000 neurons, which is huge. This is like bigger than, any of these other data sets. But there are billions of neurons in the mammalian brain, right? And so each one of these is a latent variable or confounder that is, of course, influencing, the neural activity that you're observing in the 75,000 neurons. So as part of actually the graph quilting stuff, we showed that each of these latent variables induces false positives in the graph selection event, okay? So we will estimate graphs with lots of false positives unless we deal with these latent variables. And there's some really fantastic existing work by Venkat, who actually decomposed, if we have like the graph for all neurons in the brain decomposed into the observed part and the hidden part, then he modeled this inverse covariance matrix of the observed part as sparse plus or sparse minus low rank here, okay, this assumption. And we have this estimator here, and this is just the first two terms, again, the multivariate log likelihood, and a penalty with a sparsity penalty and a nuclear norm or trace penalty to enforce low rankness. And this is really nice because it's a nice convex formulation. There's some theory on graph estimation and selection, but there's also some really major challenges with this approach. Like practically, computationally, it's really difficult to actually, first of all, it's really slow for very large data. It's also difficult to choose hyperparameters. The theory says this is graph consistent under rather stringent assumptions. And one of those stringent assumptions is low rankness. And you can show that if H, the hidden part, has billions of neurons, then this part will not be low rank. It will actually be full rank. And that's a really big problem that violates the assumptions, and it leads to false positives in the graph estimation if you apply this procedure. The other thing is a large sample complexity. So N needs to be on the order of P here to fit this procedure. So this was a really challenging approach. We tried this. It did not work out so well for the task at hand. So our solution was inspired by, of course, graph quilting, where we took the MAD-GQ estimator and we hard thresholded it. And we're like, wait a second, this is similar. Why can't we just estimate any graphical model that we want using Gaussian graphical models? Like, I don't care what method you use, graphical lasso, neighborhood, climb, tiger, whatever, and hard threshold it at value tau. And immediately, this is like super simple. It's very fast. It's very easy to understand, but you're like, there's no way that this is actually consistent for graph selection with latent variables. But it turns out it is, right? So under fairly mild assumptions. So the first one, bounded eigenvalues. Okay, that's kind of a standard one. The second assumption is the really big one here. And this is a minimum edge strength condition. And for the graphical lasso, this first term right here is the exact same minimum edge strength condition you need for sparsistency of the graphical lasso. And it's the same order. So the square root of the sparsity times square root log p over n. The additional term here is eta, and eta here is an infinity norm on the off-diagonal elements of this. And this is basically the difference between the observed portion of the covariance and the Schur complement of the effect from the latent and hidden components. And what we need is we need the maximum effect of the latent components to be bounded below the minimum edge strength. And you might say, well, this condition never really happens. And it does typically for sparse graphs and exactly the type of like disconnected graphs that we would expect in neuroscience. Disconnected, I was wrong, like connected, pathwise connected in neuroscience. And under these assumptions, you can show that thresholding the graphical lasso is sign consistent. And importantly, it's sign consistent in high dimensional settings, okay? And you can do the same thing for like neighborhood selection and climb and lots of other things. And I would argue these are much weaker assumptions than even the regular graphical lasso, because importantly, we do not have an incoherence assumption required at all to get consistency, even for recovering this with latent variables, which is a really nice approach. Yes.
I just want to come back. So you're saying that your echoes on your part, fewer assumptions, even in the presence of latent variables, than existing approaches for modeling the graph algorithm. I don't understand these. Graph algorithm is such a case.
So yes, so let me back up. It's definitely weaker assumptions than the sparse plus low rank model for latent variables. I would argue, I mean, reviewers argued with me, against me, but I would say, okay, this is a very different type of assumption than incoherence, right? It's really requiring something very different. I think in certain conditions, you can show this is a weaker assumption than an incoherence condition. There's absolutely other conditions that you can show incoherence is weaker. So it's just a different type of modeling assumption. Yeah. Okay. Yeah. Oh, yes.
Eta.
Yeah.
I can see if you assume like the number of agents is small. I mean, I think when you list that kind of skills, like you will refer number of agents in the P, right? So that's kind of the order in which data should be. But if the number of latent variables is large, why is it data to be a small quantity?
If the number of latent variables is large, why do we expect this to be a small quantity? So I think this is a good point. I mean, it really does depend on certain things and kind of how strong these are connected here. I don't think this is the perfect assumption. I do think we can like perhaps weaken this assumption to disentangling instead of a result for the whole graph on just parts of the graph and subgraph. let me answer that question also with kind of empirical stuff, which is next. And this is kind of hard to see, so sorry about this. But on the x-axis is different things we're changing. The y-axis is the F1 score, so like 1 is perfect graph recovery. And we thresholded a lot of things, including the graphical lasso, climb, neighborhood selection. Importantly, the latent variable graphical model, what you're saying is in blue right here, and the regular graphical lasso is in green. And we see that the thresholdings under lots of different assumptions, including like the number of hidden nodes going up to, this is actually going up to around 200. It starts dropping off eventually. You're absolutely right. We think that in the neuroscience example, the probability of connections is a function of the distance between neurons. So if the latent variables are really far away, hopefully there's fewer connections, and that's our hope. But I do think that this could be improved upon. It's 200. P is 200. Yeah, and so this is the same number of hidden nodes. Yeah, yeah. So, and this is a small example applied to some neuroscience data. How am I doing on time? I don't have a stop here. Okay. Oh, I see the time. Okay, great. I might skip this in the interest of time and move on. Lots of good questions. Okay, so we've dealt with the non-simultaneous recordings and the fact that kind of there were different recording sessions for graph estimation. We've dealt with dealing with the fact that there are billions of latent variables in the brain, but even just like the sheer data size, 75,000 nodes. I mean, how many people have fit graphical lasso here? Like, okay, handfuls. Okay, a couple. Like, you probably have fit them to like hundreds or maybe 1,000, but like 75,000 is just like not fun. And so computationally, first off, there's a great method, big, quick. It's like using a quasi-Newton method for fitting the graphical lasso. It's pretty fast. We tried it on 75,000 for a chain graph at a single value of lambda, and it took over a day to run. So can you imagine if we had to do hyperparameter tuning? We'd be like, tell the scientists, oh, come back in a couple of months and we'll have your estimate for you. So like this is maybe doesn't quite scale up to 75,000 nodes. But more importantly, like statistically, if you think about the conditions under which all of our graph estimates are consistent, they rely on degree conditions. And we know the synaptic degree of each of these neurons, and they're on the order of like 20,000 to 100,000. No joke, degree synaptic connectivity. So like these are super well connected neurons. And so it's very unlikely that statistically we could learn the graph for these huge graphs. So turning to a method that I've used in a lot of other settings, and that is mini patch ensemble learning. And so the goal here is to form random ensembles by double subsampling the rows and the columns simultaneously, or in other words, the features and the observations simultaneously. So if this is our original data, you permute it and you get this tiny patch. So yes, mini patch is not an error. Mini batch, you know from stochastic optimization, is subsampling observations. This is a mini patch because we also subsample features. And the idea is we're going to do this many, many times and ensemble or average the results of this, okay? So you can think of this is coming from kind of high dimensional statistics stability, stochastic gradient descent, dropout in neural network training, and so forth. So you might say, okay, well, we can sample all these little patches, and here I'm showing them as contiguous patches, but I mean, they're just random double subsamples. And what if we learned a little tiny graph on each chunk of data? And can we somehow average those graphs to get a consistent graph estimate when we have really big data? And you might say, well, no, of course not, that's impossible. But let's think about each one of these little patches first. So each patch, when we sample them, we have chunks of unobserved nodes. Well, these are just latent variables. And if this is the ground truth, we know that they're going to induce false positives. But under certain assumptions, we just discussed this. We can use thresholding to actually get rid of those false positives and get a consistent graph estimation. So the question is, what if we just sample mini patches and do like thresholded graphical lasso on each little mini patch? And why not do that and then put them together? So our approach is like embarrassingly simple. sample random mini patches all over the data, a bunch of them many times, lots of overlap, right? And fit tiny little graphical lassos to each one, threshold them, and just record the edge selection events and retain and average them and retain all of the edges that were selected frequently. And we say frequently is just greater than 50% of the time, just like really simple. And we're going to call this a graph estimate. And immediately you can see, okay, computationally, this is really fast because now I'm fitting like teensy tiny little graphical lassos instead of a really big graphical lasso. Importantly, if you've got huge data, it's actually like memory wise, it's really difficult to store a graphical lasso estimate on big data, but this is actually really memory efficient as well. So yeah, it might be that time where everybody turns into a pumpkin. Oh, okay, cool. And so statistical theory, so I'm not going into details here, but we're essentially just leveraging the latent variable stuff we did with thresholding. So the idea is we need some of the same conditions. And in the theory, what pops out is there's a small trade-off between computation and statistics. Specifically, like the larger the mini patches are, the number of nodes in the mini patch, the more likely the assumptions are going to hold statistically that we can get consistent graph estimates. But the larger they are, the larger the computational burden. And so there's kind of a sweet spot between computation and statistics that are required. Yes.
You can design a mini patches, so doesn't you recapture things like the number of patches to be better than the squared one? I guess not by regression.
Yeah. So this is a great question. So I'm going to answer your question in two parts. So we do completely random sampling of the mini patches. We always sample the mini patches so that there is fantastic sample complexity. So what we do is we tune the number of nodes in each mini patch. But we just say, okay, there needs to be like 10 to 100 times the number of samples, right? Just to make estimation of the graph on each little mini patch better. Yes.
I understand why you're saying, but I don't understand why you're saying the opportunities, because you don't really care about N for the computation of graphical assets. It's huge. Right now, you have to make a variance in the new map.
Yeah, but it's kind of like a random forest. It's a variance reduction thing by kind of.
You're going to get better results.
That's right. Yeah. It's not a computational issue. That's right. It's just a statistical issue. Okay, so let's see some results here. So my student that worked on this, Tianyi Yao, these are all of the methods in R and Python available for the graphical lasso, and they were all as of like a couple of years ago, so we might have missed some recent ones. This is our method, MP-graph, and what you're seeing in this first figure is the F1 score. So 1 is perfect graph recovery, 0 is not, and this is computational time on a log scale. And this is a high dimensional small world graph, but with P equals 500 nodes, so still like really small in the scheme of things. Our method does the best. There's a close second here. This is actually a method in Python coded up by my former student, Manjari Narayan, does really well. But hers takes almost 8 hours to run, and ours takes about 40 seconds. So to get the same statistical accuracy. And there's a lot of timing comparisons and accuracy stuff in the paper. This is just one standard benchmark, a chain graph. And what we're doing here is varying the number of nodes in the graph from 100 to 20,000. And here we're plotting the statistical accuracy via F1 score, and here the computational time again on a log scale. And you can see that the competing methods, so this is big quick, and this is also neighborhood selection. The statistical accuracy does decrease as you go down. Ours decreases very gradually. I think it has weaker assumptions, especially it does not depend on the degree of the node in the same way that the regular graphical lasso does. And computational time, yeah, it took about 8 hours for 10,000 nodes. We totally killed them because it took over 24 hours at 20,000 nodes. And so we just stopped the computation for these methods. And ours took around 30 minutes exactly to do 20,000 nodes. And I should mention this is with hyperparameter tuning of our method, and we did not hyperparameter tune the other methods. We fixed lambda. So this is with hyperparameter tuning. So on calcium imaging data, ours took about 11 minutes to estimate the graph of 14,000 neurons with hyperparameter tuning. And this is an estimate. It's a nice small world structure. There's some interesting findings here about the relations of these neurons to neural tuning, but that's for another talk and another day. And some other kind of quick related work. So Daniela, this is related to exactly what you were saying. So we were investigating this low rank covariance completion and then learning the graph and the computed covariance matrix. And there is evidence, this is 2 separate calcium imaging data sets, that there are low rank structure in the spectra of them. But there are some challenges with these approaches. They can recover certain types of edges consistent with the low rank structure, but they can't recover all edges in the graph. This is also related to a question I think, Dan, you had about uncertainty quantification. And so this is actually doing graph inference, which basically is just graph recovery with FDR control, or actually p-values and confidence intervals for each edges in this patchy setting where we have potentially different sample sizes. And this is, if you ignored the patchiness, you get a lot of false positives. And with our approach, these edges are color-coded, the width of the edges according to their p-value. And you can see it largely recovers the connected components that were in this little graph. And so this is the great work of my postdoc, Lily. And we also, Armin left, but we are talking about this. This data is totally not Gaussian, not even close. I just give a whole talk on Gaussian graphical models about data that's like super spiky. And you're probably like completely suspect about this. So we have 2 methods of dealing with this. The first one is we actually Saban graphical models are like a generalized normal. They're really closely related to a Gaussian graphical model. They're a form of exponential family graphical model. And they focus on the extremes, on modeling the extremes. And so this is a really good way to align the spikes. And we also just do some very basic like non-paranormal copula transforms, which actually in practice works pretty well for this data. So again, our goal is to take each of these innovations and eventually put them together to estimate the full best estimate of the functional connectivity amongst the 75,000 neurons that we can get. We actually do have some estimates right now that the neuroscientists are working on. Again, our goal is to compare them to the synaptic connectivity, but there's a bottleneck on the synaptic connectivity because each one of the EM microscopy slices, there's a crazy computer vision system that has to align the dendrites as they extend out through there. And so that's still a work in progress. So we actually have some estimates of the graph from the functional data. We're still waiting on the structural graphs before kind of publishing some of this work. So this is kind of a continual work in progress. It might be several more years because there's a lot going on with this data, but it's an exciting project. So just to summarize, we use graphical models to learn functional neuronal connectivity, and we highlighted work to deal with non-simultaneous recordings, latent variables, and the sheer data size using mini-patch learning. But there's tons of future directions. There's a lot of like really great statistical problems coming out of neuroscience right now, and so I think there's a lot of fun problems to work on there. And there's a ton of other extensions and methods that could be developed related to our work on each of these areas. And I'm happy to go into details or answer questions on those if you'd like. I should acknowledge, so on the graph quilting stuff, this is the work of Giuseppe Vinci, a former postdoc of mine in Gotham, Dazarathi. The latent variable stuff, Minji Wang, who's now in the job market right now, so I have to give him a plug. And Tianyi on mini patch learning, Anderson, who's going to be on the job market next year on the Saban graphical models, and Lily Zhang on uncertainty quantification and irregular measurements, who's also on the job market this year. And lots of fantastic neuroscience collaborators, especially Andreas Tolias in his lab at BCM, was super helpful in this effort. And some references, and I'm just going to stop there and take some further questions. Thank you.
Function of distance systems or that hypothesis to be. Do you think that picking the mini patch as the target physically, like small neurons would like speed up the rate at which the recovery starts?
Yeah, that's a great question. This is the second half of what I was going to answer. Erardi, did I say your name right? Sort of question. And that is like, we're totally picking mini patches at random right now. I absolutely think that you can cleverly do something that uses additional information or even adaptively sample mini patches. I think that's a really hard problem. It's something to look at. You don't just want to sample mini patches of like all the node neighborhoods together. If you can even do that, you need to shake them up somehow. So we're thinking about how best to do that. but that's certainly future work. Yes.
I had a question about the impact of learning. So just what it seems like it's only support recovery, it's not like.
Yep.
Okay.
It's only support recovery. I would argue that if you can recover the support well, you can always just go back with a constrained likelihood estimate and get an estimate of theta, right? We can't do that in graph quilting, but we can do that if we in just regular settings. Right. Yeah. Other questions? Yeah.
Certain two things are, I'm curious, obviously, like in the PhD, like 75,000 neurons, that you have these bottlenecks. Have you considered, obviously you can evaluate your method theoretically, evaluate them numerically, and simulate the data. But I imagine you could also consider applying methods to like simpler model organisms. Right? So if you go to like scale again, you can kind of get the whole thing at once, right? You don't necessarily have the challenge that you have to fill, but you can pretend like you have it. Right?
Yeah.
So have you consider doing that to see how well you can get a story of function structure coupling?
So we have considered that, but we have not done that in part because C. elegans is a different beast because the neurons do not spike. And so the neuron behavior is completely different in C. elegans versus like the mammalian brain. And so the data structure is so very different that we have not done that. But there are ongoing projects in C. elegans that have like completely mapped the synaptic structure. There's like exactly what, 200 and something neurons? I forget, 302, something like that. They know the exact number of neurons. They've mapped the synaptic structure, but that they never spike. So like mere correlation and Gaussian graphical models actually work really well for C. elegans, and you don't need to do all this crazy stuff. And the data size is not unmanageable. Oh, I think I'm just trying to say the data properties are really different. So calcium imaging on C. elegans, the data just like looks and behaves really differently. Yeah. Yeah, please.
This is very interesting results. I was wondering for these learn how much that is specific to the doctor you're looking at, and how if you have multiple
Yeah. Yeah, that's a great question. This was one mouse. They called this mouse the platinum mouse because they had previously a silver mouse, a gold mouse, and then to like get everything. But it costs like $100 million for one mouse worth of data. So they're not getting multiple mice. The idea is that if we can learn some like really basic principles from this one mouse, then hopefully we can extend this. And I should say there's ongoing efforts back to the other organisms on Drosophila also, which is not the mammalian brain, behaves differently, but they do have the full synaptic connectivity of Drosophila right now. And people are, and it's just one fly. Right? Like, so there's these efforts in neuroscience to get like one model organism set up and really analyze that and then eventually extend it. I mean, the Human Genome Project was similar. It took them years to sequence just one human, and then eventually they could do lots of others. So that's the hope, I think, with a lot of these technologies. Yeah. Other questions? Okay, dokie. Cool.